Say good evening to you. I'll turn off my camera now and share the presentation. So thanks everybody for attending this evening. I just need to change my, okay. Justin, just confirm again that you can see the screen okay?
Yep, we can see it. Looks good.
All right, thanks. And welcome again, everybody. Tonight, I'm going to be talking about mite management, especially responding to changing conditions. In beekeeping, we talk about bees part of the time, and we talk about Varroa mites the rest of the time. There's probably a couple other issues we need to talk about too, but tonight it's Varroa mite management. And this presentation, you may have seen it before, but I definitely keep updating it with new information. And so part of this presentation is how responded to changing conditions, and the mites do keep changing. I'll go through a little bit of my background. We'll talk about Varroa mite management, really what is mite management. We'll talk about what kills the colonies in winter, and I'll finish off with a treatment plan. And just as a little bit of entertainment, we've got the Beekeeping Myth-Busting series based on the popular TV series, Apivar, Bad Batch, and Resistant Mites, and also Colonies Cannot Survive Without Treatment. So my background, some of you are familiar with it. I started beekeeping in the 60s. I grew up on a dairy farm in southwest Pennsylvania. Remember my first bee hive, my dad got it. It was an AI root hive, painted it white, probably had plenty of paint on it actually. I was in the Boy Scouts and I had the beekeeping merit badge. I still have that merit badge actually. I had 12 to 14 hives as I was growing up on the farm, and those hives produced a fair amount of honey, and I sold that honey on the farm. I remember being in 4-H beekeeping, and that's where I learned a little bit about beekeeping. But I learned most of the beekeeping from an extension circular from Penn State, and also the Beekeeping Merit Badge book. That Beekeeping Merit Badge book is still good for the information that it has in it. Of course, it's missing some information. For example, we're going to be talking about viruses, and there was symptoms of sac brood, but they really didn't know what caused it. And of course, today we know that sac brood is caused by a virus. I was able to make some comb honey and sell that on the farm as well. It was a lot simpler keeping bees then. European fowl brood and American fowl brood were the main concerns. The bees needed a lot less management. It was more you put them into place, monitored them during the spring. I never really took that many steps then to prevent swarm management. I definitely collected a lot of swarms. And of course, nobody knew about mites, no tracheal mites, no Varroa mites. Start off, Colos. This was a European-based, Colos was a European-based organization studying colony losses. It's since broadened out to include the Americas, North America, Central America, South America. Now they're well represented in Colos along with Asia. So it's really a worldwide organization now. They had sent out a survey. Some of you may have actually gotten a survey. So there was a couple questions in there that were interesting to me. The first one, what factors do you think most improve the health of your honeybee colonies? Select up to 3. Well, I looked at the list, and changing queens more often, that made sense to me. Finding better apiary sites really didn't. I think I have pretty good sites, but bees adapted to local conditions, that made sense to me. Better agricultural practices, less pesticides, more diverse food sources. You know, growing up on a farm, and also having a lot of my colonies are on active farms where they're using pesticides and so forth. I don't have an issue with pesticides. The farmers there responsible users of pesticides, that they understand that pesticides can kill beneficial insects like honeybees. Better weather. Well, it seems like a lot of people always complain about the weather. My attitude is the weather's going to be the weather. I need to be adapting to the weather and be prepared for whatever it brings. So that's not a factor for me. My efforts as a beekeeper, I thought that was a big, big factor. And then this last one kind of ties into a better beekeeping education, that kind of ties into my efforts as a beekeeper. The other one, there was another big factor that I didn't see on here, so I checked that one. That was really related to question number 10. What percentage of colony losses per year do you find acceptable in your beekeeping? So that was a pretty easy answer for me, 0%. That's what's acceptable to me. I know all colony losses can be prevented. It's a mindset, really. Do I lose some colonies? I lose some that in hindsight, I know why I lost them, and I know how they could have been prevented. So the other factor really, and ties in with what we're going to be talking about tonight, is mite management. Most of the colony losses are related to mite management, either directly or indirectly. To give myself a little credibility in this presentation, I'll just share what my losses have been over the last 3-4 years now. And I don't typically lose production colonies. These are full-size colonies that are producing honey. I do overwinter quite a few nukes. I've started overwintering rather small nukes, 5-frame nukes. And I've lost a few over the winter, but I'm getting better and better at overwintering smaller nukes. There's definitely techniques to that to be successful. And most of them make it through the winter just fine. So, my overall losses, I'm going to say, are about 5%. I fortunately have a fair number of colonies, but, beekeeping is really not how many colonies you have. Whether you have 1 colony or 10 colonies or 100 colonies or 1,000, right? Beekeeping is about how healthy and productive the colonies are that are under your care. So, if you're successful with 1 colony or 10 colonies or 100 colonies, you're doing a great job. On Facebook, we see a lot of posts, and this one caught my eye. Beekeeping myth that's out there: wax moth killed my bees. Sure enough, somebody posted, wax moth killed my bees. What are these little black things? And I don't know if you can see the resolution on your screen, but this is all Varroa mites here that might have been on an IPM board. It's not wax moth, but it was kind of a palm to the forehead kind of thing whenever I saw this post. But then I looked at the date that it was posted, and it was April Fool's Day. So this guy knew what he was talking about, right? He was making fun of all those folks that said, oh, wax moth killed my bees. Well, it wasn't the wax moth, it was the mites that weakened your bees. They got so weak, and the wax moth took over. Wax moth is an opportunistic pest. And the other pest that's out there is small hive beetle. Here's pictures of both. It's kind of hard to distinguish between which is the small hive beetle and the wax moth, but the wax moth here is on the top, and I can pretty much tell by looking at its head. The wax moth larva also generally gets larger than the small hive beetle larva, but the wax moth larva has 3 legs, 3 pairs of legs here in front, but it also has this other 4 pairs of legs behind. The small hive beetle larva also has 3 pairs of legs in front, but no legs here. And it's also got these spines on the back. So if you can kind of remember, the wax moth larva has legs and the small hive beetle has the spines. You'd probably be able to distinguish between the 2 of them. So there's a lot of misinformation on Facebook. This guy, it ended up, I thought it was pretty funny that he posted this on April Fool's Day, and at least initially he fooled me. So mite management, what is mite management? Well, mite management's much, much more than just killing mites. A lot of people, think they've done their job just by knocking down the mites. Mite management's more about first understanding the biology of the Varroa mite, and we'll talk about that a little bit more. You also need to understand the population dynamics of the honeybee colony itself and the mites and the interaction. Need to understand the virus infections in colonies. We'll talk about that quite a bit. Mite management is developing a plan that includes testing and treating. It's one thing to have a plan, but you've got to execute that plan. And even after you execute it, then you've got to assess the results and develop an improved plan, incorporating any new information. And I'll share my plan with you, and that what you'll see is the result of me going through these steps myself with my own colonies. Bottom line is that successful beekeepers are successful mite managers. Another way of saying that, unsuccessful mite managers are really good mite keepers. They're good at growing mites, but not so good at keeping bees. It's been about, not last year, but year before, I was mentoring someone, and then I hadn't heard from them for almost a year. She called me out and asked me to come look at a colony because she thought it was queenless. Got there and asked her why she thought it was queenless, and she said she didn't really see much flight activity at the entrance, so suspected it was queenless. Asked her when the last time she was in the colony, and it was a while. So we opened up the colony, and we saw frames that looked like this. And a close-up picture, there was brood that was partially uncapped. I'm sorry, let me use a pointer here. Just brood that was partially uncapped. There was pupa that was completely uncapped. There was pupa that actually had its head and part of its thorax chewed away. There was larvae that was sunken down in the cells, and some of it had dried out. Here's a larva that is very dried out. So she really hadn't been treating her colonies. She got these colonies from somebody up in New York that claimed that he was treatment free, and these colonies didn't need to be treated. Now, the mites had taken over. The mite counts were very, very high in these colonies. So they're definitely suffering from parasitic mite syndrome. But I wanted to share also parasitic mite syndrome, a frame like this. Sometimes beekeepers confuse it with European foul brood because European foul brood also has a spotty pattern. You'll see larvae that has sunken down in the cell. You'll see larvae that's discolored and so forth. European foul brood is a bacterial disease of the larva. The larva food contains the bacteria, and as the larva eats the larval food, it becomes more and more infected with the bacteria. It's got to be a kind of a painful way to go. The larva should look nice and white like this larva here. Here's an infected larva. And if you look closely at this picture, here's a larva in larval jelly that is pretty white. But down here, there's a larva, and this jelly is already turning yellow, and the larva is turning yellow too. Again, here's a larva that has sunken down in the shell or cell, and you can see these white lines here. Well, those are the tracheal tubes on the larva. They stay white. Even depending on the resolution of your screen, you might be able to see some of those white tracheal tubes here in the discolored larva. So the larva typically, European foul brood really manifests itself, shows the symptoms in larva. And just to be complete, American foul brood. This is a picture of American foul brood. You'll see some punctured cappings. As cappings are sunken a little bit. Here's a capping that's partially opened up. You can kind of see the slumped down pupa in here that has died. Here's some that are, I'll say, coffee colored, or at least with cream, that have sunken down. And here's, you can see scale in this cell here. So Um, these are all brood diseases, and it wasn't really common for beekeepers to see parasitic mite syndrome brood, and they often misdiagnosed it to be European fowl brood or even American fowl brood. But really, the characteristics of parasitic mite syndrome, if you look in the top of the cells, you'll see actually little crystals, white crystals, looks like crystals of salt, and it's the mite defecation. As the mite feeds on the pupa, it'll defecate in the cell. So in the empty cells, you often see those white crystals. You'll see melted down larvae. This larvae probably died of a viral disease. You'll see brood that has been chewed away, partly chewed down brood by the workers. And you'll see what is called bald brood, uncapped pupa. And again, here's one that's partially uncapped. These are all pictures from the Bee Informed Partnership. It's a very good reference on this. So what really kills colonies in winter? Well, you know, people post, oh, a cold snap killed my colonies, a false spring killed my colonies. I really didn't even know what a false spring was until people started posting about it killing the colonies. Pesticides, you know, well, first of all, in the weather, you know, the bees have been around for eons, right? And they've been through plenty of cold snaps and false springs, and it hasn't killed them off. And also, if a cold snap or false spring killed your bees, but didn't kill your neighbor's bees, then, you know, you got to ask yourself, why did it kill your bees and not your neighbor's bees? Pesticides really aren't an issue in winter. They're not really applied. Diseases such as American foul brood, the European foul brood, chalk brood, you know, those are brood diseases, and you know you would see those manifested in the summer and the fall, not really in the winter. Small clusters, yeah, potentially a small cluster, but you know, I'm pretty amazed at how small the cluster will survive the winter if it's properly winterized. Queen, that can make a difference, but if the queen's halfway decent in the fall, they usually make it through the winter. Starvation, definitely could be an issue, but that's a beekeeper's job to make sure the livestock under their care doesn't starve. Predators could be an issue. If a bear hits your hive during the winter, could kill it, but that's hopefully rare. Hopefully you're taking precautions to keep the bears away from your hives. Most people would say Varroa kills the colonies in winter, and that's true, but that's not the main cause. Varroa will weaken the colony, but not necessarily kill it. What really kills the colonies in the winter? Well, it's these things. And you know, when I first did this presentation, it was before Corona, right? Now everybody knows what these things are. These are viruses, and a virus epidemic is what really kills your colony in the winter. You know, the Bee Culture magazine just has a recent article about Beekeeper's Guide to Honeybee Viruses, and it's a very good article. If you get a chance, please read it. I used to say there was 21, 22 viruses, and that was maybe a year and a half ago. Now there's like 37 viruses that bees can get. And just because you don't see any symptoms of viruses doesn't mean the viruses aren't circulating in the colony. Again, I think thanks to Corona, everybody's much more familiar with what asymptomatic bees that can have a virus and be asymptomatic. This is a little bit dated, this diagram here, but USDA, people would send in bee samples from colonies that are having issues, and oftentimes it's for American foul brood or European foul brood, but they collected samples and then ran viral prevalence on the samples. And, you know, they found a lot of black queen cell viruses in the colonies. That's especially worrisome to me because I make a lot of queens, raise a lot of queens. And I'm always constantly looking for symptoms of black queen cell virus. Fortunately, I don't see it. did have the colonies tested a while back for viruses, and that did not show up. Deformed wing virus is very prevalent in colonies today, probably more so than it was just a few years ago. We'll talk about it. And then there's other viruses as well. There's acute bee paralysis virus, and there's Israeli acute paralysis virus. There's chronic bee paralysis virus, slow bee paralysis virus. So all these paralysis viruses, really hard to see when you're looking at a bee, but during the winter, these paralysis viruses interfere with the bee's ability to use its flight muscles. And the bee uses its flight muscles during the winter to warm the cluster. So if they're not using their wing muscles effectively, they're not able to keep the cluster warm, and slowly the cluster diminishes. So a bee can have a large infection of deformed wing virus and not really be apparent on the outside, right? The wings could look perfectly fine, but they have deformed wing virus. But if you look inside that bee, here on the left is a healthy control, the head of a healthy control bee, showing its hypopharyngeal glands and also its mandibular glands. And here's one that's infected with deformed wing virus. So the hypopharyngeal glands produce the brood food for the workers. And here you can see these glands are pretty well developed. There's a lot of them. But over here in the one that's infected, they still have some glands, but not nearly as many. And then the mandibular glands really show up here. And in a healthy bee, there's a lot of glands. And on a deformed wing virus infected bee, again, they're very much atrophied. So, bees with deformed wing virus to varying degrees have smaller hypopharyngeal glands and smaller mandibular glands. And this results in a reduction of both worker jelly and whorl jelly. So even though the bees may look normal, they're not able to feed the brood effectively, and slowly the colony diminishes. So viruses kill the colonies. The mites weaken the colony, that's for sure. But really the issue is the viruses that the mites transmit is what kills the colonies. So how do you prevent a virus epidemic in the colony? We don't really, there really is no treatment for viruses in the colony. And, just as soon as we come up with a treatment for a virus in a colony, a new strain would come along or a new virus would come along. Like I said, a year or two ago, there was 20-some viruses known. Now there's closer to 40. And I have no reason not to believe that we'll keep finding more and more viruses that affect the colonies. Now, certainly some are going to have more of an effect than others. So how do you prevent the virus epidemic in the colony? You've got to control the mites, mite management. And part of mite management is really understanding the Varroa mite biology. And it's pretty well known. So here, the first diagram here is this. There's a mite on a bee, and this mite, and it's kind of amazing that the mites have evolved to this. It knows that it's going to get into this cell before it's capped, right? It's just kind of amazing that it knows that, right? So it jumps into this cell, buries itself in the larval food at the bottom of the cell, and it's got some structures that enable it to survive submerged in the larval food. I think they used to say there was a snorkel there, but more recently they found that it can actually close off its tracheal tubes and survive that way. Once the cell is capped, this female mite, it's already been fertilized. She lays an egg, but this first egg is not fertilized, and that'll be a male. About 30 hours later, she lays another egg, and this egg is fertilized. This will be a female. Here's that mother mite, and here's the male who's now hatched out of the egg. About 30 hours later, she lays another egg, but the first 2 eggs have hatched and feeding on the pupa. Another 30 hours later, another egg is laid. Now you have the original founder's mother mite. The male is now mature, and you have 2 immature females. 30 hours later, the mite lays another egg, and happens to be up here. Now you have both the mature male and a mature female. The female become fertilized. 30 hours later, the mite lays another egg. And now you have the mature male. He's mated with 2 mature females at this point. 30 hours later, another egg. But now there's at least 2 mites here that are been fertilized and mature, and they'll merge with the worker brood. So 1 mite goes in, and you have 3 mites coming out. This is 12 days here from the beginning to end, maybe 13 with before the cells capped. So let's say 2 weeks. So in 2 weeks, you've tripled your number of mites in this scenario. If this was a drone instead of a worker, 3 mites would, because drones take another 3 days to mature. So 3 or maybe even 4 mites would come out of drone brood. So you can see that the mite population just doesn't double every couple of weeks. It's truly exponential. So here's just kind of a simplified diagram. In the spring, the bees build up, the population builds up. And you'll see, everybody's feeling pretty good about themselves right now. Low mite counts, right? Well, the bee population's pretty high. Here's the curve for the mites, and they just really haven't, at this point here in May, they really haven't got a good start yet, right? But they'll keep increasing, and then in fall, as the bee population's decreasing, the mite population's still increasing. So now there's going to be, it's not unusual to see a lot more mites in the fall. Back here in May, think everything's fine, and a couple months later, all of a sudden the mites explode. This is from Randy Oliver's Scientific Beekeeping, and this is the rate of increase and decrease of mites. So the point of this is that the mite population, the rate of increase is very rapid, beginning in January all the way through May. And then they're still increasing, but not as quite as fast in the summer. The overall population is this red line, and it'll keep going up. You can see the exponential curve here, and it reaches to a point where the mites are so high that the colony crashes, and now there's no mites and there's no bees. So you can start off in the spring here, thinking everything is like hunky-dory. It's like a nice calm day on the beach, and all of a sudden you come into fall and you have this tsunami of mites coming at you. Another graph of the population dynamics, just to take it a little bit deeper here. Let's start off with the bee population again, this curve here. The bees come out of winter, and there's a little bit of spring loss of the winter bees, but they build up pretty steady through the summer. And then as we get into autumn, the bee population tapers off, and until finally in late autumn, it's really the winter bees that are the population. Meantime, the mites, this brown line, really just keeps building up, and it'll drop off a little bit as we go into autumn as well, because the bees are dropping off. But what the other thing that's going on is this red line is you can start off with low mites, but they're transmitting viruses to the bees, and it just keeps amping up all season long. And it really, once the bees get infected with the viruses, it just circulates in the hive. You don't necessarily need a mite to transmit a lot of these viruses. A lot of the viruses can be transmitted bee to bee. So again, Randy Oliver's has a very good mite model there. It's an based on an Excel spreadsheet, and you can put your own numbers in. So I definitely encourage you to take a look at, put in Randy's Varroa model in Google, and it will bring you to this model here. This is a model of somebody that's, let's say, treatment-free, doesn't do any treatment. Maybe it's intentionally treatment-free, or maybe they just didn't treat because they didn't get around to it. But, you know, oftentimes these colonies are very, very productive in May, June, July, and August, produce big honey crops. But while they're producing big honey crops, they're also producing large Varroa crops. And the Varroa population gets so high, and then they crash sometime in October, November, typically. And you'll hear beekeepers talk about, oh, my colony absconded. Well, it really didn't abscond. The bees died off, not in a day, but in a short period of time. And as they're dying off, they fly away, the altruistic behavior, and pretty soon you're left with very few bees. So overall, just kind of a quick review. The mite population growth, it's exponential in the spring, and the viruses increase in the bees due to the mite parasitism. Viruses are spread from brood to bees and bees to bee. At the same time the honey supers are on, the mite population is exploding. And again, you can go to Randy's Varroa model, and you can put in some of your own information and see how your treatment plan may be working. Winter bees, we're really depending on the winter bees to get our colonies through the winter. These are bees that live from September until April. They're physiologically different from summer bees. They have fat bodies in them that store a lot of the energy the colony needs for the winter, protein and other nutrients. So these, if your colonies are going to survive the winter, they have to have a healthy winter bee population. That means that they must have low viruses. To raise healthy winter bees, you need healthy summer bees. And the bees that raise the winter bees are reared in July and August. So July and August is a critical time to make sure your mite populations are low if you want to have your colony survive the winter. But remember back to the diagrams, I mean, that's really whenever the mite population's really skyrocketing, right? There's a lot of brood, and as the mite population goes up, the mites are a vector for viruses, so the virus loads in the colony are going up. The bottom line is the mite levels need to be undetectable in June and July. Now, this is probably a point here of kind of new information for a lot of people. I know from an IPM perspective, the recommendation is maybe to have no more than 3%. I'm not sure those levels really make sense whenever you put it in a context of mites and viruses circulating in the colony, that it's going to infect the winter bees. So really, to be overwinter colonies successfully, you need to have mites at very low levels in June and July. There's a lot of options for treatment. There's certainly some that's not effective. Have a red X on those. Apistan's not effective. Mites have developed a resistance to that. Same thing for check mite. Mites have developed a resistance to that. For a while, people were using powdered sugar, and that's not effective. HopGuard 2 wasn't effective. There's a HopGuard 3 out now. The trouble is that you need to apply it a couple times, and it takes a lot of effort on the beekeeper's part to make HopGuard 3 effective. And I think the jury's still out on whether even HopGuard 3 is effective. Some things that are effective, the Apivar, highly effective. It leaves no residue in the colony. You can't use it while you have the supers on, but still there's no amitraz residue in a colony. Amitraz actually breaks down pretty quickly once it comes out of the Apivar strip. So you might have some of the metabolite, the breakdown product in the colony, but the metabolite is, I'm going to say harmless or safe. Apigard is effective. I think that in practice it's not quite as effective as Apivar, because Apivar, you put the strips in. This says 6 weeks here, but you want to leave it in the full 8 weeks. But with Apigard, you have to have 2 treatments, couple weeks apart, and it's temperature dependent. Apivar can be effective too, but now you need 3 treatments with it, and there's a lot of beekeepers that just don't get around to putting a second or third treatment on of Apigard or Apivar, so it's not as effective. Formic Pro, highly effective, especially if you use the double strips. Some beekeepers only want to use a single, but the 2 strips are a lot more effective because you have more formic acid in the colony circulating around that kills the mites under the cappings. The mites really prefer the brood, the drone brood specifically. The drone brood is often on the perimeter of the hive bodies, the outside frames. And if you only have 1 pad in the colony, the formic acid vapors aren't quite strong enough to penetrate the cappings at the perimeter of the colony. 2 pads usually are strong enough to penetrate the cappings and kill the mites under the cappings. So it's highly effective. Put it on once, and then it's an option whether you even take the strips out. So oftentimes the bees will take the strips out themselves. Oxalic acid's effective only when there's no brood. I know there's beekeepers that repeatedly apply oxalic acid thinking that they're working around the no brood restriction, but I think I know that's proven not to be as effective. The bottom line is oxalic acid's only effective when there's no brood. So if you're going to use oxalic acid, actually the dribble is much safer for the beekeeper too. And I'll use oxalic acid dribble on mating nukes, for example, whenever there's no cap brood. I'll definitely use it on swarms as well, because swarms will carry a lot of mites with them too, depending on where the swarms come from. So the 3 treatments I use Apivar. Apivar, use 1 strip for every 5 frames of bees. You move and scrape the strips after 4 weeks. That's been shown to increase the effectiveness of the Apivar. Use it for the full 8 weeks, and if you do, it's 95% effective or even more. I've seen it be highly effective when it's used for the full 8 weeks. Some beekeepers only put it in 6 weeks, but that's kind of like using it you're only getting 75% of your money's worth out of it then, because you're only using it 3/4 of the time that you could. But there's a lot of beekeepers who open a package, and well, not a lot, I think a lot know, but once a package is opened, really that 8 weeks starts to count. So if you open a package and put some in one hive, you need to get the rest in the other hive, because the 8 weeks starts counting as soon as you open up the package. Formic Pro, it is temperature sensitive, and we'll talk a little bit more about some of the changes I made in the treatment plan because of temperature. But again, the full 2-pad treatment is 95% effective or more. It kills the mites under the brood cappings, and there's no residual in the honey. You can use it when the honey supers are on. Oxalic acid, it's only effective when the colony is broodless. And the other recommendation is you treat each generation of bees only once. That conflicts with a lot of beekeepers that are treating it 3, 4, 5 times, a couple days apart. The issue there is that the oxalic acid may be damaging the antenna of the bees and the queen. So you could see some bee losses and queen losses, maybe supersedure of the queen because of repeated oxalic acid usage. Yeah, the oxalic acid also changes the microbiome in the hive. Formic will too, but if you're only using formic once or oxalic acid once, seems like the colony can recover. Repeated usage probably changes the microbiome much more, depending on how frequently and what the dosage is. Then dribble is much safer for the beekeeper. Using respirator if you're using sublimation of the oxalic acid. And said you can use oxalic acid on swarms. A lot of beekeepers treat in November and December. And while that may kill mites, I got to say, if you have mites in November and December, it's kind of late, because the viruses that those mites have transmitted are already circulating in the colony. They've already done their damage. They've already injected the bees with viruses, and now they're just kind of, you can kill them, but they're kind of like they've done their job as far as causing a problem for the colony. I know a lot of beekeepers treat in November and December, and it kills some mites. From my perspective, really trying to keep the mite levels low, virus levels low in particular, I think it's a little bit late. So for my mite management plan, I have 3 objectives. I'll go through the goals and tactics of each one of the objectives, and we'll finish up with my annual plan for mite management. The 3 objectives: 1, low mite levels in the colony in the early spring. If you start out with low mite levels, it'll be an exponential growth, but the total will be much lower. If you start out with high mite levels in the spring, it's going to be a problem in the summer. Want to raise healthy nurse bees in July and August, because those are the bees that are going to raise the winter bees in September, October, and November. So with the first objective, low mite levels in the early spring. To achieve that, I want to start the spring buildup with less than 1 mite per 300 bees. That's like 0.3% or less. 0% is ideal. And no more than 1 mite in a cup of 300 bees in the alcohol wash. The issue is that in early spring, late winter, there's cold temperatures, and the colony is often clustered. What do? I put apivar strips in the colonies. I put 1 strip in for every 5 frames of bees. Usually there's between 6 and 10 frames of bees in the colony. Usually they're in the top box of 2 deep. So I can open take the top cover off, take the inner cover off, or just really lift the inner cover, and then gently place 2 strips of apivar down into the cluster. And I do this in mid-January, and then in mid-February I move and scrape the strips to improve their effectiveness. This all goes much better if 2 people working together. In other words, 1 person taking the top cover off, 1 person and lifting the inner cover, and the other person physically placing the apivar in the strip. So you can have a colony opened up and closed up in minute kind of time frame. Then I remove the apivar after the full 8 weeks, and 2 weeks before the honey supers. So you'll see this on the final plan. The second objective is I want to raise healthy nurse bees in July and August. To do that, I need undetectable levels in July and August, but there's brood present. That brood are the bees that raise the winter bees. The honey supers will be on the hives. Temperatures often above 90 degrees in July and August. So I need a highly effective treatment in June that kills all the mites, including under the cappings. For that, they use Formic Pro, and I use the full 2-pad treatment in late June. I used to do this in July, but found that there was issues with it being too warm in July. Even though sometimes I was able to find a temperature window, it was okay. But the other issue is that if I waited to July, the mite buildup was now, again, just waiting a couple weeks, the mite population can more than double. So doing it early in June enables me to have a temperature window that's better for Formic Pro. And there's also knocking the mites back to a much lower level. So the third objective is to raise healthy winter bees in September and October, November. The issue here is that, again, there's brood present, and that brood's going to be the winter bees. The issue is that there's mite immigration from collapsing colonies about that time. You know, the new term for collapsing colonies are robber lures. David Peck and Tom Seeley had a recent paper discussing whether these colonies really mite bombs or are they really robbing lures. So the colonies that are collapsing, other strong colonies will find these colonies and come in and rob them out. And the bees from those colonies sometimes drift back to the original hives, but definitely the mites come back to the original hives and their neighbors. So if you get a chance, you can do a search on mite bombs or robber lures by Peck and Seeley, and you can read this. It's an interesting paper. They did a lot of research to establish this. So you need an extended release miticide to kill the migrating mites. You have mites coming into colonies all the time in September, October, November. It's uncontrolled. You need to knock down those mites as they're coming in. And the best thing I know to do that is Apivar. Here you need one strip for every 5 frames of bees. Now, though, there's typically between 15 and 20 frames of bees in a hive, and so you need to have 4 strips in there. But if you do this, I found it to be very effective. Again, move and scrape the strips after 4 weeks, and then only remove after the full 8 weeks is up. So I try to have these strips in, let's say mid-September, and then the 8 weeks is up to about mid-November. I can come in and take the strips out, and I'll try to do a mite wash then to make sure that the treatment is effective. So mite management, it's much, much more than just killing mites. Testing is as important as treating. I know a lot of people say, well, I'm going to treat anyway. Why do I need to test? Without doing the alcohol washes, you don't know what you're starting with, and you don't know how you end it. You don't know if the treatment worked. So you'll see the timing of the treatments based on the population dynamics of both the Varroa mite and the colony. I've selected the treatments based on effectiveness, and the treatments are rotated. So I referenced Randy's Varroa model before, and again, this is what the model looks like if there was no treatment. But whenever I put my treatment plan in, so back here, if I put Apivar in, and I only called it 90% effective in January, February, March, any mites that are in the colony have been reduced, and you'll have 0 levels, but you'll have mites coming in reproducing. Treating with Formic Pro in mid-June, calling it 80% effective then. And again, it knocks the mites back down to an undetectable level. Now, here in September, October, this is whenever the mite immigration problem occurs. And this Apivar being 90% effective, it reduces it down too. So while the mites are always in the colony, you maintain low levels all year long. So this is my mite management schedule for this year. You can see I plan to put the treatments in January 19th, and then move them and scrape them at mid-February, and then take them out mid-March. And I've got enough colonies that I wasn't able to do every one in the same day, but pretty much stuck with this schedule and moved to mid-March. Then April 1st, I'm usually going around. I'm getting ready to put honey supers on. That's from a swarm management perspective. I'll try to get the supers on early. But before I do that, I do an alcohol wash on every colony. I check every colony, and I make sure that there's no colonies with high mite counts. Unfortunately, this year, I really didn't find any. And May 15th, right now, I started doing alcohol washes on colonies, and still finding low levels. By the way, Bee Informed Partnership has the Mite-a-thon on. They're encouraging everybody to do mite checks, actually last week and this week, and then send the results in. So we'll see how many I get done this week to send the results in. I'll also do another alcohol wash in June, on June 21st. And I'll also treat with Formic Pro then. If I start seeing higher mite counts now, I would I'd probably accelerate the Formic Pro treatment to have it earlier. Then after the Formic Pro's been on for enough time, I'll follow up with another alcohol wash. You know, there is a potential for a queen loss with Formic Pro. I rarely find that, but still need to verify the colony's queen right. And, you know, the honey supers are still on then. I don't really remove the honey supers until September. And I then take an alcohol wash and reapply Apivar and leave that in for the full 8 weeks. And then hopefully right around Thanksgiving, taking another alcohol wash. I say hopefully because it does get cold. It's tough to sample some bees then. But again, if you don't test, you don't know if the treatment's been effective. So mite management, again, much more treating the colony and hoping for the best. The successful beekeeper develops a solid understanding of Varroa mite biology. I hope I've given that to you, along with the seasonal dynamics of both the honeybee population and the mite population. Successful beekeepers anticipate the needs of the colony by planning for both test and treat every year. My motto is test aggressively and treat aggressively. You know, Sarah said in the opening chat about she's going to micromanage mites. I think that's what it takes. Micromanagement would be both testing and treating. Execution of the plan, you know, the plan's there, but it's adjusted based on what I get in my tests, and also any new information. I keep records of actual results, and I'm always every winter I look over the plan and see what worked and what didn't, and try to continue to improve it. Developing your mite management skills is one of the most important beekeeping skills. As I said back when I first started beekeeping many years ago, it was a lot simpler. You didn't need to know anything about mites. But if you want to have colonies that are healthy, you need to understand mite management. There's plenty of colonies out there that are living, but they're not thriving. They're not healthy. They're not really going to produce much of a honey crop, if any at all. It's like looking at sick livestock that is barely getting along. Good mite managers do have strong colonies that not only survive the winter, but also are productive the following season. So just in closing, a couple beekeeping myth-busting topics, and just like on TV, there's confirmed, plausible, or busted. I've got a bad batch of Apivar. Along with that, there goes my mites are resistant to Apivar. You hear that every once in a while because there's an Apivar failure. Here's a kind of a brainstorm list of why Apivar may fail. Everything from a bad batch, mite bombs are on here, packaging integrity, resistance or tolerance to Apivar, Amitraz, dosage isn't enough in the strips, timing of the application, and so forth. So this is kind of a list that I generated myself and listening to others about possible reasons Apivar failed. And I could eliminate a lot of the things on the list, but there was 3 things that seemed like there were good possibilities. Mite bombs, or also what's called robbing lords, was certainly one. Where you have mites, you know, you might have killed all the mites one day, and a strong colony goes out and finds a collapsing colony the next day. And literally that afternoon, they've robbed out that collapsing colony and brought back a truckload of mites to this colony that virtually had no mites the day before. Packaging integrity, I've often seen beekeepers apply Apivar that's from a package that's been open for a couple months, and it's not going to be effective. The amitraz has worked its way through the plastic strips, and it's no longer effective. And finally, certainly it is possible that there is resistance or tolerance to Apivar, or the active chemical in the amitraz. So, Frank Rekovic and I've talked a few times, and he wrote an article referenced here. He says that Apivar resistance is rare. It's been found in some commercial operations, though, but there's a wide range of resistance. Bottom line, Apivar remains an effective control and for many operations. The commercial operations where it seems like there's resistance, they're not using Apivar, they're using straight amitraz. It's a off-label way. They shouldn't be using it that way. And so in some respects, they're probably creating their own issues. What are we to do? We just put Apivar in, hope for the best. If you do that, then if there's a failure Apivar, you're really not going to detect it until 10 weeks later. Then it's too late, right? The mite population's exploded. So you need a proactive approach. So based on Dr. Frank Rekovic's protocol, I can do a test, take a sample of 300 bees, just like you would do for an alcohol wash. And then you put a little strip of Apivar in a container, count the mite drop after 3 hours, then perform an alcohol wash on that. Now between what drops from the Apivar and what you get in the alcohol wash, that's your total mites. And if you can consider the Apivar effective if 70% drop with the Apivar strip and the other 30% drop with the alcohol. It's a control failure if it's less than half. So for example, you put this together, do the test, 8 mites drop with the Apivar, you get 2 more mites in the alcohol wash, that's 10 mites altogether. 8 mites out of 10 is 80% effectiveness. So you're good to go. So you can test your Apivar this way. It's a relatively easy test to do. Last January, a year ago, I did the test, and you had to grease a little bit of a weighing boat tray here with Vaseline, put the Apivar strip in, put 300 bees in, wait 3 hours, and count the mites that fall on the pan. August that year, last year, did a much more of a test commercial operation where suspected some Apivar resistance. And so just out in the field, sampling the colonies, team of us went through, one person shaking the bees, one person scooping up 300 bees. This guy here, supervisor, oh, that's me. And then, so here's, I don't know if you can see the video playing, but. So we had about 40 samples altogether. We went to 4 different yards, collected 10 samples from each of the 4 yards. So we had 40 samples here. So that's after we're done collecting the samples. Now we're back doing the easy part was always collecting the samples, but now we're counting the mites that have dropped on this tray and then performing the alcohol wash. I use a Orbital mixer to do the alcohol wash, which I find works very well, so I can go a lot quicker through these samples with the alcohol wash. So, do we find some potential resistance there, but not terrible, that's for sure. If you think you got a bad batch of Apivar, or you think you might be resistant, you can either confirm it yourself by testing, or do determined that no, it's a myth and it's busted. So some additional thoughts on Apivar resistance. Working with this and thinking about it, we've got to realize that each colony has multiple families of mites. That all the mites aren't the same in a colony. There's a female mite that started some mites and then her daughters, but there's multiple female mites in a colony that have come from different places and aren't related. So the families aren't necessarily related. So it's not uncommon to have more of one family of mite in a colony than others. So you may have some colonies that are showing more resistance to Apivar than others because of there's different genetics, families of mites. Likewise, if you have a colony in an apiary where the mites are showing some resistance, well, those resistant mites are going to spread to the other colonies in the apiary. So those apiaries may show more resistance than others. I think for a lot of us, like say local beekeepers that do some packages from colonies with resistant mites, they'll be delivered to us. A lot of the beekeepers that are using Amitraz in off-label ways are also shipping packages and nukes to us as well. So kind of got to be careful where you get your packages and nukes from. It's hard to do though. And resistance is more prevalent when Amitraz is used off-label. Talked about that. Absolutely necessary is rotation of treatment. So even if you have colonies that are resistant to Apivar, if you treat it with Formic Pro, for example, well, now you're killing all those mites that are resistant to Amitraz anyway with the Formic Pro, and you're less likely to have an Apivar failure later on. But, definitely I've seen this manifested in a number of different ways. And an apiary is only as strong as its weakest colony. And this is part of the reason I advocate that everybody check all the colonies for mites, not just pick a couple colonies and assume the rest are the same, because you'll have a colony out there that's got a high mite load and you might not find it that way. So another beekeeping myth buster, colonies can survive without treatment. This is what we'd all hope for, right? We don't want to keep treating our colonies. Can colonies survive without treatment? So this is relatively new information. There's a behavior called suppressed mic reproduction. Involves uncapping and recapping cells. So this picture here, the A, is normal brood capping. And if you peel off one of these cappings and look at the underside of it, you'll see it looks similar to this. It's pretty uniform, matted texture. This picture here, a bee is she sensed that there's something wrong with the pupa in this cell, and she's using her proboscis and antenna to sense this pupa. This pupa's basically crying out, "Hey, I've been injured in some way." So this suppressed mite reproduction. First, the bee investigates the cell. They uncap the cells, like this picture in D here. Then it goes one of two different directions. If the infestation isn't major, let's say there's a mite in there, just the uncapping of that cell interrupts the mite's reproduction behavior, it turns out, and that suppresses the mite's reproduction. And so the cell can be recapped. And this picture F here is comparable to the picture here of B, but now you can see there's a spot in the middle where this is actual wax where they've recapped the cell. And so you can see this if you're inspecting, looking for it. It's not hard to do. It just takes a little bit of patience. The other direction this can go, and if the brood is very infected, then they'll actually remove the brood. So this behavior of brood removal is real sensitive hygienic behavior, but it's all kind of tied together. under suppressed mite reproduction. So there's a lot of work being done in this area now for bees. So yeah, it's hopeful, but it's not black or white. It's not either or. It's not treatment or not treatment, at least at this point. Fortunately, all honeybees have behaviors that reduce mite populations, certainly some more than others, like grooming, Varroa-sensitive hygienic behavior, and suppressed mite reproduction. And those behaviors can be selected. Excuse me, but still the superorganism can be overwhelmed with mites and viruses, even whenever they're doing their best at grooming and performing Varroa-sensitive hygienic behaviors and suppressed mite reproduction. So therefore, at least today, mite management is still needed, especially monitoring is required by the beekeeper. Excuse me. So just some more resources. If you'd like a copy of this presentation, you can send me an email and you can pick up on these resources. And with that, we can have some questions. I don't know if people's mics can be unmuted or how you want to handle questions. You tell me, Chester.
People are welcome to unmute themselves or post a question in the chat.
So let me, I'm not going to be able to see the chat and display this. I'll keep it up for a minute. So if someone wants to speak up with a question.
It looks like Adriana's typing you a message. Let's see. You can't see the chat right now, correct, John?
I just flipped to it, so yeah, I can.
Oh, okay.
So if we have a full crop, Adrian, what do you mean a full crop? The honey supers are full?
Fall crop.
Oh, fall crop. I'm sorry. So if you have a fall crop, when's your fall crop come in? In September, you're saying? So you might be able to delay the Apivar treatment a little bit until you get your honey supers off. I'm here, my locations, and I have locations all around the area. I don't get enough of a fall crop that it's really worthwhile for me to extract it. They certainly do bring in some honey, but I actually let that on the hive for the winter, and that just means I need to feed them a little bit less. I would get the honey supers off as quick as practical and get the Apivar on just as soon as you take the honey supers off. So Nandy says mid-August to the end of September. So you might be able to delay the Apivar to the end of September. But again, I would say you need to monitor the mites, and it's, with alcohol washes. And you may, in the big picture, you may be better to prevent the colony from dying over the winter than getting a small honey crop off of that colony in the fall. In other words, need to be thinking ahead to next year and not necessarily about the current honey crop. And there might be a little bit of a balance there. Get the supers off as quickly as you can. Landy's saying, can you do an extra Formic Pro treatment and then put Apivar in late September? I guess that's certainly possible in my mind. Part of the Formic Pro does interrupt the brood rearing, and I hate to interrupt that brood rearing when the winter bees are being generated in that September timeframe. That makes sense. So Charlie's question, is there any really point in testing? And I got to tell you, Charlie, I that the Mite Wash information always is useful information to me. You know, if I get low mite counts, that means one thing. If I get high mite counts, it means something else. At least I know where I stand. If I don't do an alcohol wash, and if I do alcohol washes on all my colonies, so I know exactly where I stand with all my colonies. I'm not guessing. And that there have been in the fall, because of mite immigration, treated with Apivar and still had high mite counts. And because I did alcohol washes, I knew I had a problem, and so I needed to retreat. And those, you know, fortunately I saw that, and the colonies all made it through the winter, and they're really in fine shape now. But last fall, it didn't look so good with the alcohol washes. So Adriana says, I had low mite counts in September, but high counts in some hives that died in the winter whenever you did alcohol washes. So did you do alcohol washes on all the hives, Adriana, or just some, and then alcohol wash the ones that were so there's another question here. Sorry, I'm trying to, there we go. I wanted to bring up my my management schedule again. So Steve is asking how often do my alcohol washes. There's 7 times, 6 times on the schedule. Sometimes I'll actually do a 7th. And you know, I often do alcohol washes with somebody helping me, and then I help them do alcohol washes, and it goes pretty quick if you have 2 people working together. And so Adrian and Landy saying that might have been mite bombed, or your bees might have found a robbing lure in September and October. That happens all the time. And you could have, let me pick a number, 10 colonies in the apiary, and only 1 or 2 of those colonies actually find the weak colony with all the mites. And those 1 or 2 colonies will end up with a very, very high mite load. And that can happen, that can literally happen in an afternoon. You know, it sometimes takes longer, of course, right? But you can have low mites in one day and high mites in another day. Have I tried the phone app that uses the camera and AI to count the mites on the frames of nurse bees? No. And I don't know how we can do that, because if the mites are on top of the bees, the mite loads are very, very high. Most of the time, the mites are tucked up underneath the right side of the bee, feeding on the vitellogenin. So they're underneath. So Joe, you're saying the camera's underneath and looking at the underneath bees? I haven't tried it. I heard it talked about, but I haven't tried it. Okay, yeah, that's a good test. Who makes the app, Joe? Okay, yeah. Now, if you find out, send it to me. I'll do a little more research. I'm curious myself. So, Charlie, yeah, while it is true that Apistan, um, um, that some beekeepers are using it, it still has some issues, right? First of all, the mites are probably going to develop resistance to it pretty quickly again because they did before. The second issue is that both Apistan and check mite are synthetic miticides that actually end up with the miticide in the wax. Unlike Apivar, that doesn't have any really residual in the hive, Apistan does. I don't recommend, even though it might be effective for the mites. It's probably not what you want in your hive. Hopefully that makes sense.
John, I was able to find the app, and I'll post a link in the chat.
Okay.
It is not well rated. A lot of people are not exactly thrilled, and it does rely on scanning from the top, which at that point your mites are a pretty big problem.
Yeah, yeah. So Sarah's asking about oxalic acid vaporization. What's the best timing? And so the best timing for oxalic acid, whether it's vaporization or dribble, is whenever there's no brood. That's really the only timing per the label. So, there's certainly beekeepers that say, well, I'm going to treat while there's brood in, but they're not following the label. I think the label says minimal brood. But I got to tell you, my colonies always seem like they have more than what I consider minimal brood in there. There's, in December. Now, I raise queens and I requeen in the fall. And so my queens are young, vigorous. They're laying eggs. And the brood in December and January, those are bees that are going to be winter bees that'll take over for some of the older winter bees that may die during the winter. Unless I have a swarm or unless I happen to have a mating nuke that doesn't have any cap brood, I use oxalic acid. I don't use oxalic acid dribble that much on mating nukes because that oxalic acid seems like it impacts the queens. I can't really explain it, but I don't have a whole lot of confidence in oxalic acid even in mating nukes. So the best time is whenever there's no brood. If I've got even a 6-inch diameter brood on two sides of a frame, that's a couple hundred bees, right? Maybe 1,000 pupa there. And if I have 300 mites in the hive, which is pretty low, those 300 mites are going to be in that brood, even though there's, quote, minimal brood in the colony. So if you got such a high mite load that you've got parasitic mites at that point in time, it will knock them off. But again, when the colony's broodless, in December and January, they're probably highly infected with viruses at that point anyway. So oxalic acid certainly is less expensive than Apivar strips, but Apivar strips are going to be a lot more effective. So when you do the calculation for effectiveness and risk versus oxalic acid, at least whenever I do the calculation, it comes out to be that I'm going to use the Apivar strips. I'm going to save money and I'm going to save bees with the Apivar strips. Yeah, and I know Landy's a bigger fan of oxalic acid vaporization than I am, obviously. Just if you're treating with oxalic acid vaporization in November, January, February, I'm thinking it's too late. You know, maybe you're lucky and you had low mites then and the viruses weren't too high, but again, the treatment plan that I have here been very successful for me over the years, and other people that follow it are able to overwinter their bees with very, very low losses.
Hey, John, I have a question. Let me know what I can ask.
Sure, go ahead.
So John, first, great presentation. I find that your schedule works really well with, well-established, overwintered, kind of full-size colonies.
Right.
When it comes to swarms, splits, I have a bunch of them right now. I'm not going to be able to probably do forming pro in the summer because they're just not going to be. They might not be, have the size need for 2 strips. I don't know exactly, if for 1 strip, what is the size? I would, So what's your recommendation if you have splits or swarms as far as your kind of how would you adjust your treatment if you want to kind of rotate the different treatments? You don't want to use Apivar every time. How would you treat small?
So the label for so the label for Apivar says you could use it twice a year. And actually, I have a lot of mating nukes that need to be treated too, and I was treating them with oxalic acid dribble, but with good, that would definitely control the mites, but I seem to lose more queens, and I hesitate saying that because I don't have a lot of data on it. But so in my mating nukes, I'll use Apivar, and then I'll keep monitoring, and then I'll put Apivar in those mating nukes in September again. So it's twice a year kind of thing. The people that make Formic Pro recommend not using it for nukes. They are testing it some though, but at this point they're saying don't use it on nukes. Again, if you have a swarm, that is a good time to use oxalic acid because there's no brood, right? You could dribble or vaporization, both are effective. Landy says that dribble, she's had bad experience with it, but a lot of other people have had good experience with dribble on swarms. And likewise, the vaporization. The issue with vaporization is the beekeeper himself being safe with it. And so a split, do you think you're going to get a honey off of these splits, Jim?
No.
Yeah. So if you're not expecting to get honey off the splits, then I'd apply Apivar. I'd apply Apivar twice.
And my second question, I think I heard you say at some point that you should treat all of your hives at the same time. That's very effective. Obviously, with splits and swarms, it doesn't really work that way. Do you obviously you gotta treat swarm, at least me, right at the bat once you get it. And then would you just kind of work with your management to get maybe to the fall time? So fall, it's when everyone gets on the same schedule at that point moving forward.
So I think I understood your question. Yeah. Now, I was really talking about testing all your colonies, doing alcohol washes on all your colonies, and Jim, you're right. I mean, I do treat my mating nukes a little different. I do treat my mating nukes differently, both from a treatment and a timing standpoint than the full-sized hives. But I do have other large nukes that are basically resource hives for me, and they'll produce some honey. So even those other resource nukes, which are really 5 frames over 5 frames, plus maybe another couple boxes on top for honey, they're on the same schedule as you see displayed here now, except that they don't get the Forming Pro. I think you can treat your swarms and your nukes or splits differently than you treat your production colonies. But I'd say whatever it takes to keep the mite levels low in all the colonies. And you don't necessarily need to treat all the colonies the same, but I think you need to do alcohol washes on all the colonies, whether they're nukes or splits or full-size colonies.
Okay, thank you.
Looks like you're still getting some people typing messages in the chat, John.
Yeah, I'm watching. That's a couple. So I'm glad that people. I know it's a long presentation. There's a lot of information here, and doing it virtually, it's hard for the presenter, of course, to see if the audience is able to follow along. So I appreciate the comments. And there is a lot of information here. Send me an email. I'll send you the presentation, and then if there's any other questions, I'm glad to answer them too. So pardon me.
I was just going to say that tonight's presentation has been one of the most well-attended talks we've given virtually so far. We had over 50 people.
Cool. So how much time does it take to do an alcohol orch a hive? 10 minutes? No way. I probably can do closer to 10 hives in 10 minutes. But Joe, I've got it down. So Joe, if I'm going into a hive to do an inspection, and collecting an alcohol wash sample was only one of the steps in my inspection. And I'm usually going through the colony, and I'm always looking for, now I'm looking for swarm cells and cups. I'm looking for eggs. I'm looking for the queen. So I'm pretty much looking at every frame. And, once I find a frame with a queen on it, I know where she is. I'll take another frame that has brood on it, take a sample from that, put it in a container for the alcohol wash, and then just set it aside. I don't do it while I have the colony open. I do it after I'm done inspecting the colony. And then I use the orbital shaker, so I can do 6 at a time. So literally for me to collect a sample, it takes less than a minute. And then while I'm doing 6 samples on the orbital shaker, I'm doing something else. And it probably takes me, less than a minute to count the mites it's pretty quick to count whenever there's zeros and ones, right? So I'm going to say, at the most it takes me a minute a hive to do an alcohol wash, but I'm doing them in sequence. to be fair. And it says, do you ever take bees for an alcohol wash from a frame of pollen rather than a frame of open brood? Yes, Landy, I do. Randy Oliver advocates that it's safer to take bees from a frame of pollen because the queen's probably not on that frame, but there's a lower probability. Sometimes she is. I mean, you're looking for her anyway. Then his thinking is that those nurse bees on the frame of pollen are nurse bees, and they'll be going back to feed the brood. So it's the same bees you get off a frame of brood. And a lot of people are afraid to take an alcohol wash from a frame of brood because a queen might be on it and they miss it or whatever. And I honestly, I tell them that taking a sample from anywhere in a hive is better than taking no sample, period. It may not be the ideal frame, but taking a sample and doing an alcohol wash is better than not doing it. And if you do that enough, you'll build your confidence and be able to get, I'll say, better and better frames. The sampling from a frame of brood or a frame of pollen is and doing an alcohol wash. It's not a perfect indicator of the mite population in the hive. It's just the best indicator that we have. So again, I think that taking any sample from anywhere is much better than doing nothing. And if you can get a better sample from a frame of brood, great. But it's much better to take a sample.
I have another question for you, John.
Sure.
I know you mark your queens. Do you mark virgin queens or mated queens?
My routine is that once I see the queen laying an egg, then I'll laying eggs, then I'll mark her, kind of thing. So I usually don't mark queens that aren't laying eggs yet, but there's exceptions. I don't know if on Facebook you saw it, had a swarm at an observation hive in a local farmer's store, right? And the owner's daughter was taking a video and went back to that observation hive about a week later with intent of cutting out all the extra cells and everything. But I was a day late because about a half a dozen virgins came out of the cells. They were still in the hive. So pulled out a lot of virgin queens from there. And as I was pulling them out, I marked them then because I had them in my hand.
Thank you.
I guess you could get a virgin. It's, the virgins, whenever they first come out of this cell, are not, they're a little soft yet. Their exoskeleton hasn't really hardened up yet. I try not to handle virgin queens too much. The other thing, virgin queens, they're awful runny and squirmy. The queen that's mated and laying, she'll sit there and be more patient while you're putting the dot on her. Other questions or thoughts or?
I think we're nearing on the end here.
Yeah. So, there's a lot of different options for treating, and just like everything in beekeeping, there's a lot of opinions about how to treat. I try to base this on as much factual information data that I had. I feel very comfortable with this plan. I'm not sell a couple of nukes, and I provide this plan with the nukes. And last year I was actually thinking about that if people followed this plan and emailed me with what the dates that they did the actual steps and what the alcohol wash numbers were, and they did this plan, and then if their nuke didn't make it through the winter, I'd give them another one the next year or whatever. But somebody talked me out of that. They said that was crazy talk, right? But that's how much confidence I have in this plan, that it works, because it works. It works for me. It works for others. So certainly there are other options, but my intent here, I really hate seeing people lose colonies over the winter. It's again, I'm coming back to an earlier slide. I think all colony losses can be prevented. I think that if you follow this plan, you can prevent your colony losses during the winter too. Landy has a question about the orbital shaker plans. What do you mean plans? I don't have any plans for orbital shaker. Just an orbital shaker. And Adriana, please ask a question not related to the talk. I'm so tired of talking about mites at this point. Everybody started listening to mites. I didn't make the orbital shaker. I bought it. Jeez, I bought it the same place I buy everything, Amazon. Adriana has a queenless hive and caught a swarm. Can I merge them? So Adriana, how do you know the hive is queenless? Is there a possibility there's a virgin queen in there? It's maybe the best way to put that. And could you merge them? I'd say yes. There may be a queen in the hive and there may be a queen in the swarm, but pretty soon there'll just be one queen. Sometimes you can put swarms in a hive and they swarm back out, but so yeah, no brood. Yeah. So a lot of people come to me for queens, but I talk them out of it because they you gave them eggs and they made no cells. That kind of indicates there may be a queen in there, though. So Adriana, today in my my cell builder, so here's a little story for you. So out in the bee yard, and all of a sudden, it seemed like a lot of orientation flight going on in front of a cell builder. Now, keep in mind, a cell builder is like packed to the gills with bees. Try to keep a lot of young nurse bees in there so they feed the queen cells, right? And the orientation flight kept getting larger and larger, kind of thing, and the bees were swarming out. Now, my cell builder, I got queen excluders on the bottom, middle, and top, kind of thing, right? So I got it on the bottom so no virgin queens can fly accidentally into the cell builder and kill all my cells. But I put frames of cap brood in there, and sometimes the cap brood has got eggs and so forth on it, and sure enough, they built a cell, and the cell emerged, and there was a queen in there. I didn't believe it at first, but went through that cell builder, and I'm shaking bees down through a queen excluder, and I finally found the new virgin queen that was in there that caused the problem. And so you can, shake the bees through a queen excluder to see if they're the queenless hive, a hive that you think is queenless. And in this case, the queen was a virgin, and so luckily I found her before she did any damage. So they drawn the you could always you could always try it. And there's a lot of things I've learned in beekeeping is from just trying it to see what happens.
Okay. Unless there are any other questions, I think we can call it there.
Okay. Thanks, everybody. You know what? I'm going to try to go back, and I'm going to just put up my email address again. It's not on that slide.
Thank you, John. This was a really great talk.
Okay, and I'll put my email address up there and just leave it up there for a little bit, and that I'll sign off if I could. No, it's not. No, I'm not. Let's see. Gotta do it this way. Sorry. Too much technology. Now it went to the end. There we go. All right, thanks everybody. I'll just leave this up here until Justin closes the meeting. Thanks again.
Thank you, John.
Bye.
I'll leave the meeting open for about a minute, and then I'll close it.
Okay. Take care, everybody.
Have a good night.
John Dodd, Town Chicago. I don't know if you called me or not, because the phone doesn't ring, or at least I don't hear it.